Today we have the pleasure of hearing from Dr. Rachel Miller, the Chief of the Division of Clinical Immunology and Merck Zimmer Professor of Immunology. Dr. Miller received her MD from NYU and completed her residency in internal medicine and fellowships in pulmonology as well as allergy and immunology at Columbia Presbyterian. She subsequently served as the director of the Allergy and Immunology Fellowship Program, director of adult allergy, and director of the Division of Pediatric Allergy, Immunology, and Rheumatology at Columbia prior to joining the faculty here at Mount Sinai. Dr. Miller's research focused on the mechanisms of the onset of asthma, with a major emphasis on the role of prenatal and early postnatal exposure on later pediatric and adolescent asthma risk. She has published widely and been the recipient of numerous awards. Please join me in welcoming Dr. Miller.
Okay, thank you so much. You can hear me okay, I hope. So we're going to be talking about why some kids get asthma. First, I can say that I really don't have anything to disclose. So we're really going to start here, actually. And so when I go to departmental grand rounds, I find that they start one of two ways. The first way is someone puts up on the screen a plot or a graph about how important their disease is from a public health point of view. And with asthma, definitely I have those slides, and I could tell you that the prevalence of asthma has been increasing worldwide since the 60s or the 70s, that it's a source of terrible or great morbidity, particularly severe asthma cases. I could tell you that a tremendous amount of healthcare dollars are spent nationally on the treatment of asthma with moderate effects. And also I could tell you that people die, including children, unfortunately. In fact, one person a day dies from asthma. So I have those slides from the CDC, but I'm actually not going to show that to you. The other way that I find people often start departmental grand rounds is actually with a case. So that's actually the way we're going to start with this particular case here. It's represented by this GPS slide, which is intentionally fuzzy for HIPAA and privacy purposes. So what we're going to be talking about is this particular kid. Actually, let me go back up to the bigger scheme. I don't have a pointer, but this is a GPS snapshot of a child who lives over, you guys are New Yorkers, so you can kind of tell, lives near the Grand Concourse, which is a heavily trafficked street. Walks to school on 125th Street. Again, lots of traffic. Spends a lot of time in school over there on the right side of your screen. Walks back, chooses to spend no time really in the green space, which is Mount Morris, which is actually quite nearby, goes back home to the apartment in Grand Concourse. So what we're going to be talking about are sort of these questions. What are the links between this kid, who turns out to have asthma? So this is a kid who was born to a mother who did not have asthma, father's medical history we don't really know. The child had a lot of early childhood wheezing, and then at about age 5 or so was developed to have allergic asthma with a high IgE, about 180 or so, and requiring medications. So the question is, what are the links between this kid getting asthma, really probably without a genetic history, and his background, his exposures at home, at school, and when he hangs out, so to speak, and how does that all happen in terms of making him at risk for developing asthma? And then thirdly, what we're going to be talking about are potentially what are the really long-lasting effects of these exposures on his asthma, as well as possibly his other health and resilience. So when we talk about asthma, like we talk about other complex diseases, there's genetics and there's environment, and clearly asthma has elements of both, right? Nature versus nurture. To get at that, I'm actually also just going to show you one slide which sort of tries to measure how much of asthma is really genetic versus environmental. So here we're looking at a slide of from a cohort in Europe, Sweden, Danish, Finnish, and monozygotic twins. So obviously twins have the same DNA sequence, right? Of course, by definition. But also I just want to point out that monozygotic twins also have the same prenatal exposures and also the same early childhood exposures, if not later exposures too. So keep that in mind. It's not just the same genetics. It's the same family and household in most cases. But nonetheless, what we have here is a slide that plots on the bottom horizontal axis, you can see the population attributable risk. right? And then you have the cumulative percent. And what you're basically seeing here is that the genetic as well as the early and prenatal environmental component to asthma adds up to about 40%, all right, compared to other complex diseases. 40% versus arguably 60%, which is really more clearly environmental. So that's what we're going to be focusing on, really that other 60%. Again, what is it about environment exposures for this kid that he's exposed to that puts him at risk? So we could spend a lot of time talking about a lot of environment exposures that are important in asthma, but instead we're going to really focus on air pollution. And in New York City, the main source of air pollution, without question, is traffic. We now know, without question again, that exposure to air pollution causes asthma. As represented here on this top slide, this is a meta-analysis of over 70 studies conducted. And here we're looking at, via meta-analysis, basically a single odds ratio, which clearly shows association from these studies with exposure, in this case, black carbon or soot, and the development of asthma in children. The same studies actually also looked at other pollutants like fine particulate matter and nitrous oxide and actually found the same thing with associations that were statistically significant meta-analysis with the development of asthma. This is a different meta-analysis. Now it's about over 40 studies, and mostly actually in Europe. And this particular study, in this particular slide, breaks down by air pollutant. Fine particulate matter, carbon monoxide, ozone, nitrous oxide. But here what we're really showing is that each of these pollutants is individually associated with asthma hospitalizations and emergency room visits. So air pollution not only causes asthma, it's a trigger for worsening asthma requiring hospital visits or emergency care. So then the next question, now that we know that air pollution is important, the question is, well, when, right? How does this happen, and particularly when are kids or even adults are vulnerable? So this is a slide published just a couple years ago that actually tries to answer that question. Is it prenatal exposure, or is it exposure later on, say, in childhood? And so here we're looking at that comparison where exposure is measured by where people live at the time of birth and throughout childhood when they access information on their yearly addresses and use that information to calculate via weighted averages exposure to specific pollutants. In this case, we're looking at NO2 and fine particulate matter, PM2.5. And we compare birth address to the current address when the childhood asthma was diagnosed. And you can see in red from this meta-analysis, again, random effects, is that it's a birth address, really, that seems to be more predictive of childhood asthma than the address and the presumed environmental exposures during childhood. On top panel, we look at NO2. Bottom panel, we look at fine particulate matter. Both of these exposures, again, are emitted from traffic sources, as well as some industrial sources in Germany, Sweden, and Netherlands. But it turns out it's really not all prenatal exposures, right? And so clearly, later on exposures do have an impact. And I think this is the best slide that indicates that. This was led by Graham Barr from Columbia. And so here we're looking at when kids immigrate to the United States. In particular, the focus was on 4 urban areas: New York City, Chicago, Miami, and San Diego. And you can clearly see here that when kids immigrated to the U.S. at a very young age, as toddlers, they're much more likely to get asthma than if they immigrate to the United States in later childhood when they're adults, right? Suggesting that there's something about the physical environment in these urban areas. We can debate whether San Diego is urban, but put that aside. There's something about the physical environment in these areas that makes it more, during that young period, toddler age 1, 2, and 3, that seems to impact or increase the risk for asthma. That's not so impactful when kids and people are older. Okay, so let's talk more about the remaining 60%. So I showed you a few slides about traffic. I'm going to show you just a couple slides about another environmental concern. Hopefully, most of you know why I have these 3 random products up here: plastic toys, thermal receipts, and the inner lining of cans, right? And the environmental concern right? Most people know, I think, yeah, maybe. You might be tripped up by the receipts, but they are all very similar in the fact that they are all essentially loaded with the ubiquitous environmental contaminant called bisphenol A and some of its analogs. So let's talk about that a little bit as well. So this is another environmental exposure of concern. This is an example of one of the studies we did a bunch of years ago where we measured bisphenol A, this chemical, again, in all these common products, in the urine of kids. And we did that at age 3, 5, 7. We looked at wheeze, we looked at asthma. And what we found was that cross-sectionally, there was association at age 5 and 7, maybe age 6, between the bisphenol level that we measured in the urine of these kids, measured by mass spec, liquid chromatography, and the likelihood that these kids were going to present with wheezing illnesses and asthma. So not only do we find that cross-sectionally, we also found that prospectively. And so here, when the bisphenol A was measured in the urine of kids at age 3, controlling for the current exposures, that was also predictive of later on developing asthma at age 5 and maybe a little bit older, age 7. Not only was the exposure to bisphenol A associated with wheeze and asthma, it also was associated with airway inflammation. So in this particular case, we measured fractional exhaled NO. For those of you who don't know, that is a gas that we emit when we have a lot of airway inflammation. And when we measure that at the children at age 7, we found that as well was associated with the bisphenol A exposure measured in the urine of kids, suggesting not only were they had wheeze, they had asthma, and they had biomarker evidence of airway inflammation, controlling for all the usual stuff that we control for in our studies. So next the question is, okay, we know environmental exposures are important. We talked to you about air pollution. We talked to you about the bisphenol A products. The next question that we were particularly interested in is, well, how does that happen, right? How does exposure actually cause disease, and particularly when we're talking about asthma? Well, obviously that's a very complicated question, and there's about 100 answers to that question. But when trying to focus it down, and in the particular case of air pollution, there are lots of mechanisms represented here, all of which have been implicated, all of which have had very solid experimental studies and epidemiological studies to suggest that they are likely pathways to development of air pollution-related asthma. What we're going to do in the subsequent slides is actually focus on pretty much the top half, going back to this issue of gene and environment and how they may relate to each other. So really, the question is not how much is genetic, how much is environment, right? That's not really the question. The question is how does our genetic makeup and our environment interact, right, to increase the risk for development of complex diseases like asthma. And so really when thinking about how that happens mechanistically, we're going to focus on the example of epigenetic regulation and how the environment or what the environment may be doing to affect how our genes are expressed and ultimately phenotype. Okay, so this is a slide of how I see the world. A baby is born, has a mom and a dad biologically, right? And is born with a genome. During pregnancy, there is exposure in the uterus and other mechanisms that we may not entirely understand to environmental exposures. For clear, I showed you that slide, right? And then the child grows up, becomes a kid, becomes a teenager, becomes an adult, and during all this period of time, the child is exposed to lots of things that you could imagine. We call that the exposome. Now, the exposome, and people who write articles about this say that there are exposome domains, general, specific, and internal. So general refers to things like, is a child brought up on a farm, or in a city, or what may be the socioeconomic status of the family. Specific may be black carbon soot, or cockroach allergen, or bisphenol A, very specific environment exposures that a kid is exposed to. And then internal is probably things like microbiome, age, maybe even stress that's within us that also contributes to just our overall environmental exposed gestalt, aka exposome. So we have the exposome. What's the exposome do? The exposome does lots of things to us on a molecular level. It affects our epigenome, transcriptome, proteome, metabolome, and probably other omes that I don't have on this slide, all of which represent what's happening to us inside molecularly and interacting amongst themselves into an all-encompassing interactome, to add another ome. And then all of this basically, through a bunch of magic and amazing mechanism, dictates whether or not a kid or an adult may have allergy, may have asthma, or may be healthy, right? And not only is it whether somebody develops asthma, but in fact maybe the type of asthma, right? Is it steroid-resistant asthma? Is it obesity-related asthma? Is it non-allergic or allergic asthma? All of that may be influenced by your interactome. So we're going to focus on the epigenome part. It's represented here. So just to focus on that, the concept is that we have environmental exposures, right? Air pollution, chemicals, diet, smoking, etc., that impact our genetic, our alleles in such a way, represented here by this red ping pong ball, that affects our packaging of DNA. Specifically, we're talking about epigenetic modifications like DNA methylation, post-translational modification of histone, non-coding RNA, microRNA, RNA methylation, DNA hydroxymethylation, and a bunch of other mechanisms actually that don't fit in that bubble, but these are the main ones that have been studied. Now, these environmental influences may induce these epigenetic marks prenatally, but honestly, it may not only happen prenatally, it may happen postnatally, or it may happen prenatally and be expressed postnatally, such that in the case of DNA methylation, for example, when that is sort of upregulated, so to speak, we get gene silencing, right? And so if you silence genes and you silence the right genes or the wrong genes, that's going to affect your phenotype, say whether or not a kid develops asthma. So here's another way of putting this, focusing more on DNA methylation, right? We're exposed to environmental exposures during pregnancy, but not necessarily only during pregnancy. Top panel, we have a diet rich in folic acid. We'll come back to that topic. Cigarette smoking, whatever it is, that induce these epigenetic marks, right? We're learning a lot about where these epigenetic marks are, what they're like. They happen in places called CPG islands, CPG shores, elsewhere through the genome, that affect the packaging of the DNA. It affects the access to things like transcription factors that are really important in gene expression. And if you affect enough genes that are important in a particular pathway, and if you're talking about DNA methylation, you silence the genes, that certainly can impact, again, downstream what's happening at the phenotype level, in this case, whether or not, say, there's airway inflammation. So that's the idea. And truthfully, if you look at air pollution, you look at the question about epigenetic regulation, there are hundreds of papers, and it's clearly evident that many pollutants, if not all pollutants, have been linked epigenetically to pathways that we know are important in asthma. And so this is a basic slide, and we're not really going to go into it, but you get the idea. Cigarette smoke, fine particulate matter, diesel black carbon, soot, and bottom row, polysaccharide aromatic hydrocarbons, all of them have been associated through a variety of epigenetic modalities to alter the epigenetic profile and hence transcription of genes that we know are important in asthma. So how do we look at this? Well, this is one of the ways we've been looking at this. This is our cohort from Columbia called the Columbia Center for Children Environmental Health. I showed you the Bisphenol A slides came from this cohort. And so here we have just sort of a depiction of what we do, that we are measuring environment exposures during pregnancy, but also during childhood. A variety of them, air pollutants such as polysaccharide aromatic hydrocarbons, soot, smoke, chemicals, etc. Variety of biomarkers repeatedly during the course of childhood. And we've been following these kids now who are reaching age 18, 19, 20 for various outcomes. And I've been mostly interested in the asthma and the airway inflammation outcomes. We recruited 727 Dominican, African American women during pregnancy in northern Manhattan and the South Bronx. Established in 1998. So what are some of the early things we learned? So I want to show you this slide, which dates back a while. But one of the things we learned is that the kids in our cohort and the mothers in our cohort are exposed to a lot of air pollution. All right? So this is an example of how we measure it during pregnancy. The woman wears this little backpack for 2 days. And there's an inlet that goes near her mouth, and you can measure what she's inhaling. right? And we've used that to measure polysaccharide aromatic hydrocarbons, which is another traffic-related air pollutant. And we found that they're all exposed. And all the babies appear to be exposed as well. And in fact, if you measure something called PNA DNA adducts, which is actually not only a measure of exposure to pHs, but actual DNA damage, some people think it's a marker of pre-neoplasm, that was found in 40% of the cord blood samples. When we followed the kids as they get older, all the kids had evidence of exposure to pHs in their urine. But it's not just pHs. We have exposure to other air pollutants, vanadium, fine particulate matter, black carbon. These not only are big exposures, but they've been associated in our kids with cough and wheezing illnesses and decreases in lung function. And all of this seems to vary by season, by how high the floor is where somebody lives, whether they face a street versus an alley, and other factors. So I'm going to show you a little bit of work that we've done with the pollutant black carbon, otherwise known as soot. You guys all know what that black stuff is. So this is a component of fine particulate matter. It's a traffic emission. It's an industrial emission. It's that black stuff that comes out of the smokestack in many, many buildings nearby. We know for years that exposure to black carbon, and I showed you that early slide from the Environmental International publication, meta-analysis, that's associated, that black carbon exposure is associated with asthma development. It's also associated with asthma symptoms. So these are all basically facts at this point, but what was really unclear was how this happens. What's the time course? Is this acute? Is it chronic? Are there lag effects? And the underlying mechanisms have always been uncertain. There's a belief that black carbon is chemically inert, and all it really does is serve as an adjuvant for airborne allergens, and that may be true or partly true. But there was also some recent speculation that this may be an exposure that's epigenetically regulated. So we worked on that hypothesis, and we asked whether acute exposure to black carbon would in fact be associated with DNA methylation of genes that we know are important to asthma. We measured DNA methylation in kids, and by getting to, we didn't get to the lungs of the kids, but we actually collected DNA from their buccal mucosa, which is not so far away, and exposed the air pollutant. We were particularly interested in the question of lag effects based upon other literature, and we were interested in the question what was the role of allergic sensitization in all of this. So for this particular study, I'm going to show you what we did. This is kind of a snapshot of our protocol. We were focusing on kids now aged 9 to 13, all right? So pre-tween teens, and you know what they're like, right? So we go to their home, and we hook them up with a device, I'll show that to you, that allows us to measure how much black carbon they're exposed to every 5 minutes for a 24-hour period of time. We would put a sampler in their home that measured residential exposure to other pollutants, such as vanadium and particulate matter. I'm not going to show you that data. And we came back 5 days later, and we assessed them again. We did spirometry to measure lung function at the beginning and at the end, and we collected buccal DNA at the beginning and the end so we could look at short-term acute changes in DNA methylation, as well as look at some of the time courses that may be happening over the course of a week. And we repeated all of this 6 months later so we could begin to look at the impact of season. So this is kind of what the kids wore, except this is the original version of the vest. We actually upped our game and have a sportier one now, which is from Patagonia, and they liked it better. Although on the right side we have the halter, design that actually was designed by FIT grad students and was more popular among the women in the summertime. I don't know. Anyway, so they wore a vest, and inside the vest we had what's called an air monitor. You can see it there in the middle there, about the size of a Sharpie, that was sewn inside the vest, which was hooked to a little cable here and an air inlet near the mouth, so we would know what air was being collected, and that was what we used to measure black carbon soot exposure. In addition, we had a GPS device in the kid's vest, and I showed you some of that GPS data, so we knew where the kid was going. And we had 2 ActiCals activity monitors. We had one on the wrist, so we would know where the kid, again, was walking and how active the kid was, which we looked at in other questions. And we had one hidden inside the vest to make sure that the vest and the kid were moving together, right? Because, 12-year-olds, right? We don't really know, we need to know they were actually wearing the vest. And this is how we found out whether they were wearing the vest, and actually they were. So I showed you the bottom panel before of one of the kids in the study, and you could see he walked along these heavily trafficked routes to school, went home, didn't really go anywhere else. That's on a Tuesday. Upper panel is on a Thursday. And one of the striking differences is that the exposure, the amount of exposure to black carbon is completely different from a Tuesday versus a Thursday. In fact, it was double on a Thursday. For reasons that are unclear, but you could suspect that looking at his GPS, and we're calculating this, he spent more time on streets that we know had a lot of traffic. Okay, and so when looking at evidence for epigenetic regulation, we see this is one example of some of the findings that we have. We focus on genes that we knew were already established hotspots in terms of DNA methylation. Not only candidate genes, but areas, again, that had previously been pinpointed as being important. So we focus on, for example, this allergic gene, interleukin-4, and we see evidence here of decreased methylation decreased methylation in association with higher level of exposure to black carbon, right? So the idea being that more air pollution, less methylation, more expression of this pro-allergic gene is what this may indicate after controlling for all the usual things that we need to control for. Not only did we see this, we saw it actually 5 days later, really, not the same day, suggested of perhaps a lag effect to some extent. We saw this with other genes as well, NOS2A or INOS, which is another pro-inflammatory pathway, unclear findings with interferon gamma. In this particular bottom panel, what we're looking at is association between lower methylation, presumed increased expression, and higher levels of this biomarker of airway inflammation, fractional exhaled NO, as evident repeatedly and on average among these kids, and also after controlling for things like season. Not only do we see that, when we actually look at the kids and we characterize them by whether they were allergic asthma versus not allergic asthma, here you can see that there actually were differences. And in fact, it was really the kids that were known to be allergic because they had a high total IgE, but in particular, they were allergic to cockroaches. Those were the kids that really showed this effect whereby the evidence of epigenetic regulation and their pro-allergic genes, the cockroach allergic kids, more so than the kids that were allergic to other allergens. So that seems to be a group of kids that were particularly susceptible. We looked at more genes. This is FOXP3. For those of you who don't know, FOXP3 is a transcription factor that's really key in T regulatory pathways. And one of the many hypotheses or pathways about asthma is that there's impairment of T regulation that allows for extra inflammatory processes to be going on. So here we in fact see that when the methylation is higher, there's presumably more silencing of the T regulatory gene, right? So is inflammation going awry, so to speak, or to exaggerate? And here we see evidence that that is associated with decreased lung function measured on the spirometry when we went to their homes. Okay, so in summary, what we're seeing is that exposure to black carbon or soot may be inducing epigenetic marks. In this case, we're focusing on DNA methylation of genes that we know are important in adaptive immunity and the pro-inflammation and allergic sensitization profile in the kids that seems to be associated with airway inflammation. We know from other studies that it may not be black carbon soot, it's other pollutants. It could be Bisphenol A or other bisphenols. We know that allergens are important as well. In our particular study, cockroach seems to be the one that is most impactful in some of these pathways. Okay, but what we also know is that this is a profound oversimplification of what happens in asthma for many reasons, obviously. Most important that kids are complex. As I showed you earlier, there are many pathways involved. We're looking at buccal DNA, which is not exactly what's happening in the airways, and on and on. This is just a simplified model. So with that, we knew that we needed to interrogate more in depth what may be happening mechanistically to really understand these processes. So that leads us to the third part of this talk is what more is really happening mechanistically, what really can we pinpoint And then the other question on the bottom of the screen is, what are the really long-lasting effects? I showed you earlier that we know that prenatal environmental exposures are important. Can we study that? Well, you can't really study that very well in a kid who's 11, 12, or 18, but you certainly can study that in mice. So we decided to move this operation, so to speak, into mouse models, right? So you take a mouse, we take mice, and in this particular case, the pollutant that we were looking at were the polycyclic aromatic hydrocarbons, I mentioned before, traffic-related air pollutants, and we exposed wild-type mice to this aerosol either during pregnancy or when the pup was born for its early life. We did a very typical mouse model where we sensitized the mouse to ovalbumin, and then we did what's called an airway challenge. So the airways were sensitized, and we looked at things like airway hyperreactivity and how reversible that was with the beta agonist procaterol. We looked to see and ensure that the model worked, and there was upregulation of IgE. We collected cells from bronchial lavage in the mouse, and we collected tissue from the lungs now to look at DNA methylation of genes and pathways that we were particularly focused on at the time. So this is another way of describing our approach. On the upper right is a picture of our chamber. We can put 3 mouse cages in there. So they run around and do their mouse thing. And while they're doing that, you can see there the nebulizer. It's exactly the same nebulizer you see in the emergency room. And through that, we expose them to a mixture of polycyclic aromatic hydrocarbons versus negative-controlled air, so 2 chambers lined up at the same time, during the course of study. And then there's a place there where we can pull out a filter and we can make sure that the pHs were aerosolized, and we can measure that in the lab, or, well, my colleague's lab. Okay, so the idea is being that we can assess genetic influences because we looked at biopsy mouse strain, which is a very inbred mouse strain, but it was chosen because it's known to have a high affinity allele for the aryl hydrocarbon receptor, which we know binds the pHs as ligands that we're studying. We are able to assess environment because we delivered very specific controlled environmental exposures. The pHs that we delivered were actually the same, proportionally the same to the pHs that we were measuring amongst our pregnant women who were wearing that backpack for 2 days that I showed you. So we tried to mimic what was happening with the pregnant women with our pregnant mice. And then we were focusing on epigenetic regulation, and we were looking at differences in known hotspots, canon gene methylation at the time, of genes of interest to us, at least that was the start, and the functional outcomes that we focus on were airway hyperreactivity, airway reversibility, and IgE. And not only that, we actually were looking at the offspring, so we were looking at transgenerational effects, and actually in other studies we were looking at grand offspring. Okay, so I'm actually not going to show you the results. I'm not going to show you the results because unfortunately they were actually not particularly compelling. And we had a little of this, a little of that, p-value was 0.05. We managed to publish in open journals, which was okay, but profoundly disappointing. You know, we thought this was going to be future for the next 10 years, but truth of the matter is it really wasn't. So we were sort of stuck with a situation where we thought we had this brilliant model where we could study lots of things, get grants for life, and we found out that maybe that wasn't quite true. So we were debating what to do, right? And this is sort of the pause part, you know, for the learners in the room. You know, what do you do if you have this hypothesis that you just know is great, right? And it turns out not to be so great. It's wrong, it's correct, there are study design issues that you can't fix. You know, what do you do about that? And so that's where we were at this point. And then we sort of took a step back and thought, well, you know, we have a great model, but maybe it's not a great model for airway disease. Maybe it's a great model for something else that could also be affected by air pollution. So in other words, maybe not everything is about the lung. It's a bit painful because I am a lung doctor, but actually it's probably true in life. And it turns out that coincident with this, our cohort at Columbia, CCCH, was simultaneously looking at other complex diseases related to air pollution exposure. And here I'm showing you some fabulous epidemiological work that showed that prenatal exposure to the pHs was clearly associated with obesity in these kids at age 5 and 7. Robust epidemiological work. Impressive, right? So we thought, okay, obesity. What about our mice? So we had weighed the mice, of course. And here's the thing. our mice were actually heavier. Now these are biopsied regular wild-type mice, and the only difference between the groups is the pH exposure, but when we looked at their weights, and actually calculated the mouse BMI, which is not on this slide, both the males and the females had evidence very early on of being heavier if they were exposed prenatally to the PAHs. Not only were the mice heavier, their adipocytes were bigger. As represented here by the slide, this is looking at brown adipose tissue. We found the same thing when we dissected out white adipose tissue. And not only were the adipocytes bigger, but there was evidence of altered gene expression in genes that are important in adipogenesis. The genes that we looked at were COX-2, PPAR gamma, fatty acid synthase, and adiponectin. Gene expression was higher. So that was interesting, and because I guess ultimately, not ultimately, but because of my interest in epigenetic regulation, we of course had to look at that. And here's an example of one slide looking at the gene PPAR gamma, which is an important slide in adipogenesis, but it's also a gene that's important in lots of things, including tumor suppression and growth and so forth. There's a lot on this slide, but the bottom line is in both males and females, brown adipose tissue and white adipose tissue, every single time, not every single time, every single CPG that we examine, on average, there was evidence of the pH-exposed, prenatally exposed mice having decreased methylation. Okay? Suggested to presume increased expression. And not only did we see this in the offspring mice, we actually saw it in the grand offspring mice, okay? The babies of the babies, suggesting that there was something happening across generations, both the obesity phenotype, the increase in size, the adipocytes, the relationship between the increase in size and the gene expression, and the methylation, and the phenotype of weight gain. So we thought that was pretty interesting, and we were a bit excited, and we thought, let's just keep on going. And so we did. We also had work from our cohort that not only was air pollution associated with obesity, but air pollution appeared associated with neurocognitive issues, ADHD, for example. Let's model that, and so in fact we did. And here's an example. This is an open field setup, which honestly I hadn't done since I was senior in college. But you put the mouse in a chamber, and I'll spare the details. But basically, we're looking at mice that were prenatally exposed either to the pHs or controls by the algorithm that I showed you. Males versus females. And what you see is that the mice that were exposed to the pHs prenatally, they tend to hang out in the periphery of this chamber, kind of hovering down and basically pooping a lot, which for a mouse is evidence of a stressed out mouse. And for reasons I don't know, that was for the females, not the males. So the female mice are stressed out, apparently not the males. Not only do we see this phenotype, when we actually looked at gene expression and promoter methylation, we found evidence in the cortex of these mice that key genes, in this particular case, GRIN2B, which is a gene important in excitatory neurotransmission, which is important in things like memory and learning, and another gene called BDNF, which is important in dopaminergic pathways and mood disorders. These two genes showed evidence of altered gene expression, and in the case of BDNF, where we looked, evidence of altered methylation. That was negatively correlated with gene expression. So that proved to be also quite interesting. And then one day I get a call from a colleague of mine, epidemiologist, who says, I hear you have this great chamber that looks at all sorts of diseases with prenatal exposure to PHs, and I would love to do a study with you on breast cancer. And I'm like, I'm too busy. I'm an asthma doctor. Leave me alone. But you know, the other two grants didn't get funded, but this one did. And in fact, we were able to look at mammary tissue outcomes, same exposure system in the offspring, represented by F1, and the grand offspring represented by F2. And what we found is that when we look at the mammary tissue, when we look at it under a microscope, we see evidence of the prenatal pH-exposed mice having more mammary cell hyperplasia, as well as more evidence of proliferation, epithelial cell proliferation, which are biomarkers or precursors of neoplasia. Same mice, same biopsy mice. Not only do they have this phenotype, they have evidence of altered expression in the middle panel and DNA methylation of pathways we think are important in breast cancer pathogenesis. Here we're looking at the gene estrogen receptor alpha, but we found this for other genes in the pathway, aryl hydrocarbon receptor repressor, BRCA1, where gene expression in the mammary tissue was decreased in association with prenatal pH exposure in the offspring, and again, the grand offspring. And that seems to be associated with altered DNA methylation as evidence in the bottom panel there. Okay. But, you know, I'm still an asthma doctor, and I can't wait to get back to the asthma researcher, because looking at adipocytes on the microscope was really never my thing. So going back to taking all of this and thinking about, again, these primary questions: how does a kid living in a city get asthma when it doesn't appear to have genetic risk? We need to revisit that particular topic. And so fortunately, we're able to do that through a new study, which is called the Children's Respiratory and Environmental Work Group, called CREW. I don't know if we have any rowers in the room, but the idea is that we have 12 of us, 12 birth cohorts across the United States, have come together as a single asthma consortium. The first time in the United States this has happened. Very common in Europe. This is the first US cohort to do this, where we take our data, we pool it together, we harmonize it, hence the rowing. If we work together, we can move faster, to ask some of these fundamental questions about what causes asthma and how this may happen, which we can do so much better as a consortium than we can as a single site. This is the basic premise of CREW, that we pool our data together as best we can, and we're looking at elements related to symptoms, related to lung function, demographics. We're looking at specific exposures, allergens, chemicals, pollutants, and how that may affect whether or not a kid has asthma, and more importantly, what type of asthma, what the actual phenotype is. Then we're also looking at the mediators involved, particularly that lend to the asthma endotype, and that again brings us back to the epigenome, the transcriptome, are the two areas we're really focusing on in this cohort. And this is an example of what you could do when you team up. I know it's hard to read, but the point being that if you can see or imagine the horizontal axis, each of them refers to a different biospecimen, right? Blood RNA, IgE, nasal lavage, all of which we've been doing in these kids, or in the process of doing these kids right now, and pooling together to answer some of these fundamental questions about asthma. I'm just going to show you one slide so far, but we have a bunch of papers circulating around. We have something about to come out in Lancet Respiratory that I'm really excited about, but I'm just going to show you this one to give you an idea of what we can do, right? And this also gets to the question of, this is actually really tough work to get all these groups together and figure out even a basic question of how do you define asthma. This is a paper that addresses that, where we have these different definitions of asthma in different colors here, and depending upon which definition you use, abnormal spirometry, wheeze on exam, history of wheeze in the last 12 months, depending upon what you use, you actually get different data results regarding how common asthma is in your cohort. And this is a representation of that. Top panel, we were looking at cohorts that meant to reflect the general population. That includes Columbia group. Bottom cohort are cohorts that were recruited because we thought that there was we knew there was a family history of allergy and they were more at risk. One of the interesting observations is here is that our cohort was not recruited for at risk for asthma, yet you see how skewed we are to the right and how much asthma we have, right? So that's New York City. Thank you very much. So, going back to the remaining 60%, this is actually what we can now start to address, not just in our cohort, but much more nationally. Looking at pollutants and my additional interest in the phytochemicals, how that measured across the country, different types of environments, rural, we have rural Wisconsin there, urban, New York City, different socioeconomic groups, how we can look at those factors in relationship to environmental exposures, in relationship to the epigenome, transcriptome, and how that may impact the asthma phenotype. So we're really excited about this work. We have 4.5 years to go, but it's already really exciting, and that's really our main direction. But for the purposes of this, I added one more element here. Hopefully I've convinced you that air pollution exposure is bad for asthma, so are other chemicals. These things are ubiquitous. And sometimes when we give these talks, people say, well, I kind of knew that. I can't do it, but so what, right? So air pollution is bad. Okay, well, people try to live in less polluted areas, but they may not be able to. Cigarette smoking is bad. Of course, we all know how difficult it is for people to get quit. So what do you do with that information? What's the so what? And people sometimes say, well, I can't do anything about that, so why bother? And I would disagree with that perspective. So part of so what, it's not only understanding what's going on and trying to identify kids at risk, but part of it is, right, to intervene, to do something. So here's an example of approach that one group has taken, Dr. Zhang and actually Baccarelli, when he was at Harvard. And so this is focusing on the exposure of fine particulate matter, right? So the premise that they're going with is that these air pollutants induce inflammation systemically, locally. This inflammation is responsible for oxidative stress, and that's responsible for upregulating pro-inflammatory pathways, particularly T helper cells. I agree with that. And that is responsible for alternating the DNA methylation landscape. So if you could intervene against that, you could fix things, right? And so they asked the question of, can we fix the DNA methylation landscape by giving people who are exposed B vitamins? Seems simple, right? So they designed a pilot study. It's just 10 adults healthy. Single-blind crossover study. There's a sham exposure, then there was 2 hours of pretty intense exposure to fine particulate matter. 4 weeks after that, daily vitamin Bs that are known to be methyl donors, all right? Folic acid, I showed you earlier slides on that, B6, B12. And here what you're seeing is evidence of, before and after, that there clearly were genes who exhibited evidence of DNA methylation that was attributed to the administration of the vitamins among the fine particulate matter exposed volunteers. And in this particular slide, we're looking at methylation patterns in the blood, specifically the CD4 positive T cells. That's vitamin B. This is vitamin C. So this is another study, epidemiological study, where pregnant women were recruited, and they were asked, did they smoke during pregnancy or did they not? Needless to say, you can't do a randomized study on smoking during pregnancy, and the intervention was vitamin C. Why vitamin C? Well, vitamin C does many things, probably, but the idea is that it's a scavenger for all those free radical products that are products of oxidative stress, scavenger them up, and maybe things will be better. So we have the non-smoking group, the placebo group, vitamin C group, and what you can see in these select genes, they were selected out, it's not all the data, but you can see again, evidence for some genes that the methylation is basically corrected, presumably bad methylation in the placebo group, both the placebo group and the vitamin C group are smokers. So presumably the adverse methylation is reversed by giving vitamin C in these select genes. Placenta, cord blood, and buccal cells, the cells that we've been working with in the other studies, seem to also show evidence of effect. So maybe you can do something. Now, the other reason why this matters is because we need to really think about the fact that his commute may actually get worse. Or not his commute to school, but maybe this kid's commute will get worse due to changes in our environment. This example of climate change. I won't be political, but clearly we know that this is going to be impacting respiratory diseases, asthma, and allergies. And basically exposures that this kid or other kids may be exposed to in the future. And that's a very global issue, obviously. But not all of it is global, right? Some of it is actually right here. And so I just think about, well, what's going on here? What's going on here meaning 96th Street, is legislation on congestion zone pricing, right? So this is something about our community in New York City that may be impacting exposure to air pollution for our kids, right? So this began in April at the cutoff. And if you think about it, the intent was to decompress traffic below 96th Street, which may be a good thing. But that may lead to more traffic congestion above 96th Street, which probably is not a good thing, particularly if we think about a lot of these kids that are otherwise very vulnerable for air pollution-related diseases. So not just global, it's actually very local when we think about these things. And when we think about it, as we learn more about air pollution, it makes us wonder, is this a good idea or is this a bad idea? And I actually don't know the answer to that. But if we know more about what air pollution is doing, maybe in time we would know the answer to whether this is a good idea or not when we think about the health of kids and other people in our community. So I'm going to stop there, and thank you very much. Lots of people helped. I showed you lots of stuff over years. Upper panel are the people with crew. Special thanks to people in my lab that were there who took this journey with me to the east side to come to Sinai, so thank you very much. That's the end of the talk, and I can take questions.
You focused on urban areas for obvious reasons, but I want to talk about rural areas. A number of years ago, babies raised on farms were exposed to all kinds of things, of course, and their incidence of asthma was actually decreased, as if they had become tolerant on exposure probably to allergens. Now, I'm wondering if there's been a change in the incidence of asthma now that chemicals are so widely used in rural areas on farms. And I was thinking also about the forest burning in Brazil as they clear land, and the outback in Australia where it's just so hot now that it spontaneously erupts with fire.
Yeah, thank you for that question. And you're spot on. It's been known for many years that there's a farm protective effect. There are many mechanisms that have been demonstrated, including epigenetic regulation, but that may not be the case. There's also evidence that it's exposure to endotoxin. There's evidence that it's actually exposure to unpasteurized milk. But to answer your questions about chemicals on the farm, which is a fabulous question, the one study that I could think of that kind of addressed that was a comparison. It's an England Journal of Medicine paper. I can email it to you. Compared the farming community of Hutterites, Dakota area, versus old Mennonites. Did I pronounce that correctly? Right? And the old Mennonites had more modern farming techniques than the Hutterites, which is very, very traditional. And this group, led by Kara Olber and another talented people, was able to show differences in the prevalence of asthma by these very distinct farming communities, and they're also very inbred. There's not a lot of mixing, and the presumption. Well, first of all, and that seemed to be associated with various differences in gene expression, some very interesting biomarkers suggesting the different pathways that were impacted by these different limit arrangements. But the presumption was that the cause of these differences was related to the type of lifestyle and the way people farmed, where the old Mennonites were a little bit more modern in their approaches and used more chemicals, again, presumably, not demonstrated, put them at greater risk for asthma and actually allergic asthma. So you're right, not all farms are the same, for sure. And yeah, I didn't get into the whole issue about the wildfires. The situation in the Bay Area and California is a huge asthma trigger. It's something that I hope to be working on in a project that we're designing out there as well. So there's a lot of other environmental exposures of concern, now and in the future. And we may not be doing better at it. We may be doing worse. Although some of the exposures are actually improving.
Thank you very much. Thank you very much.
Okay, thanks.